to this symposium sponsored by ESTOR on endotoxic shock from pathophysiology to advanced targeted solution. I'm co-chairing this symposium with John Callum, and we also have him speaking together with Silvia De Rosa, who is one intensivist in the group of Vicenza, Italy, and has been part of the International Renal Research Institute of Vicenza. So with no further ado, I think John Callum does not require any introduction in the world of critical care and critical care nephrology. That's why I asked him directly to start his speech on gut dysbiosis and endotoxic organ crosstalk from endotoxemia to endotoxic shock. John, please.
Thank you, Claudio. It's a pleasure to be here. I assume that you can see my slides, or you'll tell me otherwise.
We do.
Let's see if I can advance them here. You may be wondering what this term gut dysbiosis is, and certainly if you went to medical school or graduated from medical school before about, call it 2013, you probably never heard this this term while you were in training, and certainly it's a term that has come into clinical use and in the literature over the last decade. So as we understand more and more about the gut microbiome, we understand that there's dramatic changes that can occur over the lifespan of humans as well as other animals. And in critical illness, there's significant disruption of microbiota. And basically what happens is that this imbalance of the gut microbiota results in a loss of beneficial input from beneficial organisms and expansion of pathologic microbes. And this has resulted in really an explosion, as you saw from the previous slide, of research in this area affecting all kinds of disease processes. This includes everything from cardiovascular disease to dementia to a metabolic syndrome. What we're going to talk about today, however, is the effect of this change in microflora on the permeability of the gut. And so as you lose beneficial bacteria that maintain a healthy mucus layer, the mucolytic bacteria reduce the mucus barrier. They destroy junction proteins and result in increased translocation of bacterial products, most importantly being endotoxin. In this study, the animals were gut dysbiosis was induced, and then a high-fat diet was given, and in this setting, these mice would develop metabolic syndrome, whereas the same diet resulted in no metabolic syndrome in animals with a healthy gut flora. So this has significant effects, but this endotoxin translocation has far-reaching implications, not only for the health of the organism over the long run, but also short-term implications. Now, you may be wondering how you can get endotoxin into the bloodstream and not develop endotoxemic shock. Well, part of the explanation for this has really been around for some time. Going back to 1946, it was discovered the concept of endotoxin tolerance. And although we understand more about this concept now, we still don't fully understand all the mechanisms responsible for endotoxin tolerance. It's clear that there's a relationship with endotoxin load and immune system activation. When these are low, the macrophages and the complement activation can be controlled. And this is a condition of tolerance where we don't develop shock and we essentially downregulate the macrophage response. This has potential to not be entirely beneficial, however, because we can have reduced response to infection. And so people that are sort of chronically leaking endotoxin into their bloodstream and downregulating their macrophage response can actually be at risk for developing infection. But as the endotoxin load increases and immune system activation increases, we can reach a point of uncontrolled macrophage and complement activation. And this can result in cytokine expression, sometimes to massive levels, as well as microvascular thrombosis. And the result of this, of course, is shock and multi-system organ failure. Now, as an aside, recent evidence suggests that these same pathways are stimulated by other molecules, a vast array of molecules which interact with not only the toll-like receptors, but other external and intracellular receptors that induce essentially the same response. And there's a host of these. I've simply listed some of them here, but they fall into two broad categories: damage-associated molecular patterns and pathogen-associated molecular patterns. And of course, the most important pathogen-associated molecular pattern is endotoxin, but it's not the only one that can induce this response. Now, humans and other mammals have a host of really three separate signaling pathways and detection systems for endotoxin. The toll-like receptor, particularly toll-like receptor 4, but also there are other mechanisms to detect circulating endotoxin and intracellular endotoxin. And these induce similar or at least overlapping pathways of inflammation in the cells. Importantly, humans appear to be exquisitely sensitive to endotoxin for reasons that are still of, I would say, controversy as to why this is the case. It may have something to do with our meat-eating diet, but we are much more responsive to endotoxin than other meat eaters. Amongst the other primates, and this is a comparison to macaques, you can see that of the top 100 regulating genes, endotoxin induces a much more rigorous response. This is a log-fold change. It's a much more rigorous response in red than you see from the macaques in blue. Now, importantly, endotoxemia is not equivalent to bacteremia. Endotoxin in the bloodstream does not equate to primary or even secondary bloodstream infections. Greater than 70% of patients with sepsis and high endotoxin activity actually have negative blood cultures. And of the 30% with positive blood cultures, only about half of those are gram-negatives. Endotoxemia can result, of course, from active infections of gram-negative bacteria. Some antibiotics release endotoxin as they kill the bacteria, but the predominant source of endotoxin in the bloodstream in both chronically, as I've showed you, but also in critical illness, is loss of barrier function and leak of endotoxin from the gut. And this also includes COVID-19, which has been shown to compromise gut function and result in gut barrier function and result in translocation of endotoxin. The gut has more, it's estimated to have more than 1 million times the lethal dose of endotoxin. We all carry around enough endotoxin in our guts, it purified and injected intravascularly to kill an entire population of Pittsburgh. Now, endotoxin and endotoxemia are not the same as, or endotoxemia is not the same as sepsis. There's about 1.5 million people in the United States alone who develop sepsis each year. About 20% of those develop septic shock, and about half of those develop endotoxemic shock, septic shock with high levels of endotoxin. And endotoxic shock is associated not just with cardiovascular system failure, not just with with shock, but with other organ failure. And the overall mortality rate exceeds 35%. In some studies, it exceeds more than 50%. Now, interestingly, the distribution of organ failure is interesting, and in particular, the difference between sort of what I would consider to be organs that concentrate various molecules, including endotoxin, such as the liver and the kidney, the organ system that is most affected by intravascular abnormalities, the complement and coagulation systems. The lung is less clearly affected by endotoxin compared to other organs. This distribution of organ failure that we see is actually quite interesting. If you use machine learning, as this study used, to separate patients with sepsis into 4 phenotypes, and I won't go into all the details about how this was done, but the minority of patients, about 13% of patients, have this pattern of organ failure that includes high levels of acute kidney injury, hepatic dysfunction, and endothelial dysfunction, resulting in an extremely high hospital mortality. And in fact, the 90-day mortality can exceed 50% in these patients. So there is a subset of patients with sepsis, somewhere between 13 and 20%, call it, who have a very aggressive disease process that may be associated with endotoxemia. Now, what do we do about this? Well, of course, one approach, of course, is to remove the endotoxin using endotoxin absorption. And you can take this treatment, which involves the use of a column which has polymyxin B that's covalently bound to polystyrene fibers. And the result of this is that you can demonstrate where this has been used extensively. For example, in Japan, this is a study comparing more than, looking initially at 42,000 patients, finding 4,000 or so that were treated with polymyxin, and then using propensity score matching to look at a control group of patients. And what these investigators found is that the 28-day survival was substantially improved with the use of PMX, an absolute risk reduction of about 6.8%. As the organ failure, in a separate study, as the organ failure increases, the efficacy of the effectiveness of the column seems to increase as well. Again, this is observational studies. If your organ failure score is low, for example, using SOFA less than 7, you have no statistically significant effect of the treatment. As it increases, you develop a small effect, and as your organ failure increases more, you have a significant increase in the effect of the therapy in terms of improving survival. Now, in studies done in the United States and Canada, this is a study that looked at use of polymyxin B in a slightly different way. It focused on the use of endotoxin activity as a way of identifying patients with high levels of endotoxin in their blood and targeting those patients. And although this study overall was negative, even in the subgroup of patients that had a high MOD score, so they had sufficient organ failure and they received 2 treatments of the therapy, we're really only able to demonstrate about a 3% absolute risk reduction, and this was not statistically significant. However, a post hoc analysis of these data, and it is a post hoc analysis, indicated that there was about 17% of the patients in this study that had levels of EAA that were above 0.9. And this equates to a potentially astronomical amount of endotoxin in the bloodstream, perhaps much more than could be treated with the column. And so if these patients are removed and you just focus on the patients with sort of a treatable high, but treatable level of endotoxin in their bloodstream, you find a significant benefit for PMX compared to sham. And this is statistically significant in terms of survival at various time points. This finding prompted the current trial, which is currently underway, the Tigris trial. And this is a study in the United States, including 15 centers that are randomizing patients with this level of endotoxemia and significant organ failure to receive PMX or standard of care. Now, this trial would already be finished if it wasn't for this guy, and of course, this is the SARS-CoV-2 virus, and of course, this, we all know how this has decimated not only populations around the world, but it's also decimated our ability to conduct sepsis trials in various countries, including the United States. And I, as an aside, I just point out that this is an interesting disease process, because the COVID disease results in significant inflammation in some patients, and this appears to be the mediator of organ failure. And when this occurs, it's really fair to think about this as sepsis. This is viral sepsis, but it's sepsis. However, the COVID virus can also affect the gut directly. And when this happens, there is evidence that there is an increase in endotoxin leak from the gut with subsequent organ failure as a consequence of endotoxin. And of course, this magnifies the effect of systemic inflammation. And there's very good evidence now. This is some data from Thailand. And what these investigators have shown is that 98.3% of the endotoxin that, or of the patients who have endotoxemia in COVID-19 have bacterial DNA in their bloodstream. And the vast majority of this are proteobacteria species, and so very much likely to be coming from the gut. And as an aside, if you treat these patients with polymyxin, you find the same sort of pattern. If patients have extremely high levels of endotoxin, you really have a difficult time bringing them down. Whereas if patients have a more manageable level of endotoxin, it is possible with the therapy to bring it under control. And so in conclusion, I would say that gut dysbiosis may predispose to endotoxemic shock, that low-level endotoxemia may reduce innate immune response to infection, but also the compromised gut barrier function may be magnified by illness resulting in endotoxemic shock. There's an uncontrolled activation of macrophages and complement, which further injures organs. And there's strong rationale to relate endotoxemia to organ failure and death in sepsis, and endotoxin removal has been safely practiced for decades. EAA levels between 0.6 and 0.9 appear to identify a target population that can be treated with PMX, particularly if it's limited to 2 treatments 24 hours apart. Increasing organ failure, for example, by use of SOFA scores greater than 9, is associated with increased effectiveness of the therapy and a much easier time demonstrating the efficacy of the therapy. And with that, I thank you for your attention.
Thank you very much, John. A quick comment before I give the word to Sylvia. I remember at the beginning of COVID-19 pandemic that I was reporting to you the fact that we had several patients with superimposed bacterial sepsis. And in fact, we treat them with endotoxin absorption, hemoperfusion, and so on. And you were a little bit skeptical at the beginning, and we advanced the hypothesis that this could be due to dysfunction of the barrier at the intestinal level. Do you recall this? Have you seen after in the subsequent, let's say, waves, similar phenomena?
Well, I think we probably all regret some of the naive ideas that we had back at the beginning of the pandemic. And I have to say, as usual, you were right that these patients have endotoxin in their bloodstream, and we and you and others have demonstrated that this is in fact the case. I continue to see patients in my clinical practice as recently as a couple of weeks ago who really seem to be kind of in a stable single organ failure. It's not getting better. They're still, on high levels of oxygen, often young patients sort of stuck in the ICU on high levels of oxygen on the ventilator, but it's single organ failure. And then suddenly something changes and it looks like they develop septic shock. And, we treat them with antibiotics. We lose many of them, I must say. And I think that some of these patients, we often never grow anything from the blood. I think some of these patients actually develop late gut barrier dysfunction and endotoxemia, and that's the mechanism of shock in these patients. There are some studies, including the one that I showed you from Thailand, but also some work that we've done with investigators in other parts of the country, parts of the world, including the US, of documenting the levels of endotoxin in these patients. And so I believe that it is a function of, it is a mediator of shock in these patients.
Well, thank you very much. I think that the presentation of Sylvia that will certainly include some of our common experience. As you know, the Vicenza model describes a perfect integration between nephrology and intensive care, and this is the case at our hospital. And Sylvia will basically speak about the clinical management and the role of polymyxin B hemoperfusion also, in light of the fact that she has been collecting data for the registry here in Italy that we're trying to put together to better understand which one are the patients that really present major benefits from the use of hemoperfusion. So Sylvia, please present your talk.
Thanks, Prof. So I want to start with a historical milestone. So we know that in 1676, we have a discovery of microorganisms. And after, in 1892, we discovered endotoxin, and it was Robert Koch and his colleague that demonstrated that purified endotoxin injected intravenously is lethal to laboratory animals. And we arrived until 1994 with the polymyx-bound hemofilter remove endotoxin. And they discovered that can be removed endotoxin with extracorporeal circuit for the circulation, and it can rescue patients from endotoxemia. So is the cytokine storm relevant in septic shock? Yes, because inflammation is a necessary evil. So from one side, we have a pro-inflammatory response characterized by leukocyte activation and infiltration, complement activation, coagulation cascade activation. From the other side, we have anti-inflammatory response with apoptosis of T, B, and dendritic T cells and increase regulatory T cells. So we start from an insult. We have triggers, PAMPs and DAMPs, and we have sensors. So we have pattern recognition receptors, such as toll-like receptor. And from a link between endotoxin and toll-like receptor 4, we have as an immediate result a cytokine storm. And so LPS is one of the most potent immune activators that gain prominence. Liberation LPS from bacterial cells after antibiotic-induced bacterial cell lysis can evoke a multitude of toxic biological reaction causing deterioration in some circumstances and adverse effects promoting septic shock in these patients. This is important because when we use antibiotic, we can also increase endotoxin release. For example, piperacillin can increase a really high level of endotoxin if we compare with other antibiotics. And what we know that during an episode of severe endotoxemia, concentration of both lipopolysaccharide and its lipid A course fraction are liberated from gram-negative bacteria and become elevated within the systemic circulation. We know that polymyxin B has a profound binding avidity for the lipid A course fraction of lipopolysaccharide, and we know that the mechanism of this binding avidity involves the development of attractive force between the cationic groups of polymyxin B and the anionic groups of the lipid A core moiety of lipopolysaccharide, and also we have hydrophobic interaction too. This is important because beyond peak concentration hypothesis, as Professor Ronco teaches us, we know that we can apply blood purification therapy such as high volume, high filtration, and cut-off membrane CVVHT modality, but at the same time, we can go straight removing endotoxin using hemoperfusion. So we know that just in 1964, as we can see in this editorial published on JAMA, endotoxic shock was such a really huge problem, but also a therapeutic dilemma, because at that time, despite a variety of available procedure, no technique exists for removal. Now we are 2020, and we know that close to antibiotic therapy, close to source control, we can apply endotoxin removal, and we can do straight an immunomodulation, removing cytokines too. So adsorption is an extracorporeal process in which molecules dissolved in plasma or blood bind to the membrane structure or to other dissolved substances, I mean resins or gels. And we know that what is important concerning adsorption is the device adsorption capability. So the total quantity of a specific molecule that the device is able to adsorb. And also important is the selectivity, because it's a safety parameter. We have in comments a lot of devices able to perform adsorption. Some are able to do adsorption convention, such as Oxiris, and so able to remove cytokines too. But how we can understand the level of endotoxin? There is a chemiluminescent bioassay, the endotoxic activity assay, based on the oxidative burst reaction of activated neutrophils to complement coated LPS IgM immune complexes. And what we know is that if we have a range from 0.6 to 0.9, we have endotoxic concentration 1,000 to 4,000, and it can correspond to endotoxin burden. That mean amount to endotoxin extracellular fluid volume around 12.5 and 50 microgram. So according to the endotoxin dose response curve, an EA value of 0.9 corresponds to 4 nanogram per milliliter. So if we consider adult patient with EA of 0.9 and a blood volume of 5 liters, the endotoxin burden could be around 20 microgram, but we have to consider interstitial space. It is a volume of 13,000 milliliter. And the endotoxin activity of 0.9 is equal around 52 microgram. So if we can see the device and the device adsorption capacity, so polymyxin B hemoperfusion is really high. And considering that the endotoxin activity, say over 0.9 is equal to endotoxin burden over 52 microgram. So in around 1970, polymyxin B was discovered to be protected against endotoxin-induced hemodynamic shock, but at the same time was extremely toxic for the kidney and central nervous system, so was not easy to use for this reason. So since 1994, polymyxin B has been bound and mobilized to polystyrene fibers, and the use of this device was able to remove endotoxin, so interrupt the inflammatory cascade, and also to decrease the excessive systemic host inflammatory response to infection. At the same time, it is important because we can also try to recover from immunoparalysis in sepsis. So in literature, we have a lot of randomized control trial from 2005 and 2019, but we have also 2 data registries. So UFA study was a really important Italian study, a randomized and blind study of 64 patients. And the results of this trial were the absolute risk of death at 28 days improved significantly from 53% in the conventional therapy group to 32% in polymyxy BHP treated group. But the trial was not blinded, and the endotoxin activity assay was not measured. And the population was a population with intra-abdominal source of shock. The Hebdomix randomized control trial is a French randomized control trial, and eligible patients were randomized to standard care versus standard care plus polymyxin B hemoperfusion. 28 days mortality were 28% for polymyxin group versus control group and was not significant. But the trial had a lot of limits because they had a lot of technical issue. I mean, cartridge clotting and failure rates. And in the same time, the observed composite mortality was substantially less than the estimate included in the sample size calculation, thus decreasing the power of the study to detect a difference. Euphrates randomized control trials we saw before show no difference in 20 days all-cause mortality, but the trial showed that in some patients with septic shock, the burden endotoxin activity was extreme because in this trial was measured endotoxin activity assay and was over 0.9 in this case. So an amendment to the protocol was made after the interim analysis that suggests a lack of effect in the patient with MOTS of 9 or less, and those patients were excluded. In a post-hoc analysis, authors evaluated the impact of polymyxin use in patients with septic shock, and with the endotoxin activity assay measured between 0.6 and 0.89. There was no significant reduction in 20-day mortalities, but when they adjust the mortality was at 28 days less than polymyxin HP group versus sham. So when we saw our study, we have concerning the design, really huge differences concerning prescribed dose, concerning the timing, concerning the results in hemodynamic, length of ICU stay, survival. This is important because meta-analysis showed that among adult patients with sepsis, septic shock, the use of polymyxin B hemoperfusion compared to standard therapy was not proven to reduce 28-day mortality or to reduce organ dysfunction scores. The problem is that we have a lot of trouble. Yes, we know that polymyxin B hemoperfusion treatment could improve the immunosuppression state of the septic patient. And the mechanism of action should be studied, and also the results of systematic review with the meta-analysis could not give a definitive answer. So we need also a still randomized control trial targeting the predefined adequate patients who are likely to benefit from polymyxin B hemoperfusion. But overall study suggests a significant survival benefit that we have a lot of literature. What we did in our center, we have a group, we have a really defined critical care nephrology group, and we use this flow chart. So when a patient comes with the high suspicion of septic shock and endotoxic shock, we perform endotoxin activity assay, and if endotoxin activity assay is over 0.6 or less than 0.6 plus positive blood culture, in 6 hours time, we confirm the presence of endotoxic shock, and if it is like that, we start with polymyxin B hemoperfusion, a 2-hour treatment. After 24 hours, we repeat the same treatment. If patient has renal failure and needs renal replacement therapy, we continue with a sequential extracorporeal therapy using membranes that we need. we have in our center. So critically ill patients with the sepsis-induced mods require complex articulated therapeutic approach. And so in 2020, we can start to talk about sequential extracorporeal therapy, meaning to restore organ crosstalk. Because if I use polymyxin Hemoperfusion to remove LPS, in the same time, I go straight on target trigger removal, and I can perform immunomodulation, so prevention LPS-induced release of TNF-alpha from macrophage. In the same time, I go to target removal, removing cytokine storm that we just have, and perform immunomodulation and avoid a multi-organ failure. So we can tell that this is a adjunct, but also we can perform organ support therapy bridge to SUS control and antibiotic therapy to improve multi-organ failure and patient outcome. In COVID-19, probably cytokine storm is nothing more than a tempest in a teapot, but what we know is that the critically patient with COVID-19 with ARDS had the circulating cytokine levels that were lower compared with patients with the bacterial sepsis and similar to other critical ill patients. Probably severe COVID-19 is associated with deep and sustained multiphase cellular immunosuppression. And we know that, as we saw before, that the SARS-CoV-2 spike protein binds to bacterial lipopolysaccharide and boosts pro-inflammatory activity. So we can have direct infection of the mucosa, decrease antibacterial defenses, increase mucosal permeability, bacterial translocation, and systemic leak of endotoxin. And we can have this kind of patient superinfection. So in case of COVID-19 infection, with the suspected or confirmed superimposed gram-negative bacteria infection, we suggest to use, as suggests also this panel or expert, polymyxin B for 2 subsequent days. And if cytokine release syndrome is present, this treatment should be followed by methods to cytokine desorption if organ support is required, CRT. So we need trial, but we have also to registry, and the data on registry could be really important. We saw here, we collect from the first wave cases that we perform and we're collecting this registry during first wave of pandemic COVID-19. And what we saw was a decrease of SOFA score, a decrease of lactate levels, also in decrease of vasoactive inotropy score. We applied this flowchart, so we suggest for this kind of patient too. And those are our data following this flowchart. As we can see, the source of infection could be 44% community-acquired pneumonia, so it's not just abdominal source of infection. And the SAPS-2 score of this patient was 71.50. So not so low. And vasoactive inotropy score in this patient decreased from T0 to T120 after treatment, also lactates. And the ICU mortality was 13.1%, and the hospital mortality was 38%. And this is the decrease of endotoxic activity and also procalcitonin. In COVID-19, we have similar results. Patients were 10 of these 32 patients. And we have also a decrease in endotoxin activity and also procalcitonin. So my conclusion, in a comprehensive approach to endotoxic shock treatment, we have to evaluate kidney function, vasopressor requirement, endotoxemia together with SUS control, and also antibiotic therapy. We want to do a dynamic monitoring and prescription because we want to achieve to precision medicine. At the same time, even a more specific treatment shall be provided for every moment that patient has a particular need during ICU stay. So we strongly suggest to use polymyxin B Hemoperfusion, but in a target population. And we strongly suggest to use our flow chart and to have a really good team to perform this kind of treatment. And thank you.
Thank you very much, Silvia. And we have just 6 minutes for discussion. I would like to ask both of you if you think that the ideal randomized control trial will ever be conducted. And in this case, what are the perfect endpoints? Is mortality the right endpoint, or maybe we should look for different endpoints? Let's start with John.
Well, I don't think we'll ever be able to abandon randomized controlled trials in medicine. I think they provide the strongest source of evidence, and so it's going to be very difficult, I think, to move away from that. I think where the real problem is that we researchers and industry have got into sort of a bit of delusional state, thinking that the way to solve negative trials in sepsis and acute kidney injury are to enroll larger numbers of patients. And what this results in is diluting any potential signal that we find. And so I think, the studies that are currently being done, now, of course, I'm biased because I'm involved in these studies, but I mean, the current studies that are being done with PMX involve carefully selecting patients and trying to demonstrate effectiveness in a small subset of patients with very clear targets, having high endotoxin and having high organ failure. I think that's the place where randomized controlled trials would be beneficial. If you try to do randomized controlled trials on huge data sets with patients or huge numbers of patients where, some of the patients really are sort of in the gray zone, you're going to just have a negative study, and that's not going to help move things forward.
Silvia?
Yes, I think that we have also selected centers because this kind of treatment require experience, and I think the flowchart probably can help also registries just to select the really specific center to perform this kind of trial, because probably sometimes could be helpful to do this kind of evaluation.
I remember when we discussed about some intervention in the past, considering patients not too sick, the possibility to see any effect was absolutely impossible. On the other hand, if you consider patient too sick, the level of severity is so high that the effect again is not visible, and you have to identify the intermediate severity. This is typical approach of identifying the correct patient that may have benefit from the therapy. So patient endophenotyping will probably be the solution for this. And today, what is the typical patient outside any randomized controlled trial that you would like to treat? And do we have specific biomarker beside the measurement of endotoxin activity assay? John?
Yeah, so I think it's a very important question, and it probably will take more than 2 minutes to address properly. But I think that if you think about the downstream implications of high endotoxin levels in their blood, they're not just clinical implications of shock, et cetera, but there's activation of macrophages. Do we have a panel of biomarkers to measure for macrophage activation? We do. They're just not clinically available in most countries. We can't get them back quickly. So being able to characterize that, I think, is much more important. We also see a lot of complement activation in high endotoxic conditions. And of course, we can look at complement and use a combination of clinical measures. Ferritin has become a very important biomarker in COVID, but ferritin is also very much elevated in situations where there is macrophage activation syndrome in sepsis, for example. And so you can get ferritin levels back quickly. They're it's a certified lab test. So that's an interesting measurement that can be applied to estimate the degree of inflammatory burden, and that may be a role that we can play. Claudia, I'd like to give you, since I'm the co-chair of this session, I'd like to give you a question and let you finish us out. One of the things we haven't spoken about, and maybe Sylvia wants to chime in as well, is the timing issue, because that's also an extremely important, situation, in the sense that if we treat too late, we're probably not going to be beneficial either.
Yeah, I think that Sylvia can confirm what I'm going to say. And this is the fact that as soon as we see a patient that is suspect for sepsis, we start the measurement of endotoxin activity assay. And we do dynamic monitoring because we do repeated measurements so that we do not wait until it is too late. And this is probably the key of starting in the right patient, also because you can see a trend in the concentration. Sylvia?
Yes, fully agree with the compliment, fully agree with the golden hour, because I strongly believe that if we start early, we can, have a really good result concerning outcome of these patients. And probably this could be the focus, and this is what we want to show with our data in the future, just to, give value to our flowchart and validation of this flowchart.
Yeah, just to close this session, which has been very interesting, I would like to remind that these therapies are intended to be preventive and protective, not a cure for sepsis. So, in other words, we need to catch the patient at the right moment and try to avoid what is the downstream consequence of complement activation, endotoxin activity assay, distributing in the circulation, and so on. With this, I would like to thank the speakers, my co-chair, John Callum, and speaker, and Silvia De Rosa from Vicenza, Italy. And I'd like to send all you back to the mainstream program of the Vicenza course on AKI and CRRT. Thank you very much.